We started the ALS CURE Project in 2019, and it came out of a unfortunate experience I had with my dear wife Gretchen, who in 2016 was diagnosed with ALS, and we fulfilled a promise to her to work towards a cure for ALS so that nobody else had to suffer her fate. The ALS CURE Project, along with our research council of clinical researchers, that is the mechanism that we use to identify which research projects to fund. Many of them were presented from the lab, and we'd present to the council and we'd select those that would get funded.
We had the great fortune of being introduced to the ALS CURE Project and meeting Mike Piscotti through the Livermore Lab Foundation. So my team and I had the opportunity to attend two-day symposia where we got to learn about ALS, what the challenges are in the current research field, what the obstacles are and where we might be able to help apply our technology, and importantly meeting experts in the field with whom we could then start to talk about projects and collaborations.
We received subsequent funding from DoD, Department of Defense. It's called a CDMRP program, and we have received funding to do what is called drug repurposing, trying to perform drug repurposing using data from patients from VA. And it's grown into not just computational, it has grown into other areas of the lab too, including trying to develop biomarkers for ALS, and now leading into microbiome analysis in ALS. That's an effort that's just going to start.
TDP-43, kind of from the ALS perspective, it's like the TDP-43 aggregation is seen in I think 93% or 97% of all ALS patients. So it's pretty much everywhere. This is the hallmark of ALS, and we need to understand like what makes it aggregate. They need kind of molecular level understanding to kind of know more about its mechanism, and that's where we come in.
What we do is we raise funds. We have events, and we also get private donations, and some of them come from the home campaign from here at Lawrence Livermore Lab, which we're very grateful to have. The ALS CURE Project is we've organized that we're all volunteers. So we are able to turn 98% of every dollar that comes in around and goes directly to ALS research.
Through the ALS Foundation, Livermore was funded to kind of look at the molecular characteristics of TDP-43. And this is hard to do because TDP-43 is a protein that has intrinsically disordered parts, so meaning that it doesn't really have a defined structure. So, and then that also means like when they come together, they can kind of interact in multiple different ways. And this is actually important for ALS because the TDP-43 proteins can aggregate. And in ALS patients, you see these large aggregates of TDP-43 proteins.
We had a major effort in developing a three-dimensional brain-on-chip. The engineers developed a way to record the function of neurons in three-dimensional space. As a biologist then, we developed ways of forming these very complex neuronal networks within this device. Developing an ALS-relevant neuronal system on our device was really the next logical step, and that's what we're working towards now. We are not ALS experts, but we're learning, and we're really relying on our collaborative partners to bring that expertise to the project.
So what we're trying to do is trying to develop causal machine learning techniques to look at longitudinal electronic health record data that we have received from VA and trying to tease out medication that can potentially prolong the life of ALS patients, which also means slowing progression of ALS. The interesting aspect of this is the drugs are already FDA approved. They are off-target drugs. They were not designed for ALS. By looking at this kind of longitudinal data of ALS patients, we are seeing whether some of those drugs can potentially be useful for ALS. Out of this thousands of drugs that we have, that ALS patients routinely take, we have been able to flag a handful that are promising repurposing candidates.
We are still in the early development stages of our ALS platform, and we are poised to take some baseline measurements to understand how these ALS relevant cells behave in our organ-on-chip system. So those experiments are currently ongoing. So once the baseline has been established, then we can start to evaluate therapeutics and drugs. So stay tuned.
Now we're trying to kind of bridge the scales. We're trying to see that, okay, what can we learn from the lower resolution models and kind of transfer that into progressively kind of coarser and coarser models that we can then refine. And the idea is to look at the whole progression of where TDP-43 kind of has its normal function to where its function breaks down and it over-accumulates.
I'd like to thank many of my colleagues who are working on these research projects here at the lab, and they're incredibly bright and also very empathetic people that know the importance of the work that we're doing.